Some women can see 100 million colors, colors you and I will never see, not because their eyes are better, because their DNA is different. Your eyes contain 3 types of color receptors, 3 cones. Every shade you've ever seen, every sunset, every painting, every forgotten face, all of it, built from signals sent by just those 3 receptors. But somewhere out there, in a very rare population, a small group of people carry a 4th cone, a 4th channel of color, a whole extra dimension of sight that the rest of us can't imagine. Let's start with the baseline. Inside the retina of your eye sit photoreceptors called cones. You have 3 types, one tuned to red, one tuned to green, one tuned to blue. Your brain takes the signals from those 3, blends them in real time, and paints your entire visual world. This is called trichromacy. About 99% of humans have it, and for a very long time, scientists assumed that 3 receptors was the absolute ceiling. It wasn't. In 1948, a Dutch ophthalmologist named Hessel de Vries proposed something quietly radical. He had been studying colorblindness, specifically the mothers and sisters of colorblind men. The women in his clinic weren't colorblind themselves, but something about how they perceived color was persistently unusual. So de Vries proposed an idea that at the time sounded almost like science fiction. What if these women were carrying an extra color receptor? Not a standard red, green, or blue cone, but a 4th cone, tuned to a wavelength sitting quietly between the standard 3, sensitive to a thin slice of the visible spectrum that the rest of us simply do not register. That idea sat on the edge of plausibility for decades. Most scientists were skeptical. The number of cone types, 3, had been settled science for over a century, but the hypothesis refused to die. In 1993, 2 geneticists at Cambridge, Gabriella Jordan and John Mollon, confirmed that de Vries had been right about the genetics. Some women, and only women, were carrying the genetic blueprint for a 4th cone, in a significant fraction of women worldwide. But genetics alone wasn't proof of function. It took until 2010 for Jordan and Mollon, using a custom-built test specifically designed to require 4-cone discrimination, to confirm the first woman whose brain was actually using all 4 receptors. Her existence had been theoretical for 62 years. Now, finally, she was real. Here's why that matters. 3 cones let you distinguish roughly 1 million different shades of color. That's the world you and I live in, 1 million. 4 cones, in theory, 100 million. A 100-fold jump in perceptible color. Think about that for a moment. A tetrachromat can look at a single green leaf and see 12 distinct shades of green where you and I see 1. A sunset isn't a gradient to her. It's a stack of separate ribbons, each one distinct. The shadow of a tree isn't gray, it's a tapestry of subtle blues, ambers, and lavenders. A private universe of color hidden inside a small number of people, walking past you, looking at the same world, and seeing something you can't. To understand why tetrachromacy mostly shows up in women, we need to talk about a chromosome, specifically the X chromosome. The 2 genes that build your red and green cones, called OPN1LW and OPN1MW, both live on the X chromosome, side by side, just a few thousand DNA letters apart. Men have 1 X, women have 2. That single fact changes everything. Now, occasionally, one of those color genes mutates. The mutation shifts the cone's sensitivity along the visible light spectrum, not by much, just a few nanometers, maybe 2, maybe 7, just enough to retune the receptor to detect a slightly different wavelength than the standard. In a man, that mutation almost always causes color blindness. He only has 1 X, so if his single X carries the shifted gene, his perception is permanently downgraded. Reds and greens start to bleed into each other. About 1 in 12 men on Earth lives with some form of this, making it one of the most common genetic conditions in human biology. But in a woman, something completely different happens. If one of her X chromosomes carries the mutation and the other one carries the standard version, her retina ends up expressing both. 3 normal cones plus a 4th, slightly shifted one, side by side, both fully functional. The same genetic accident that downgrades a man's vision can upgrade a woman's. It's not a trade-off, it's not a price paid, it's pure biological asymmetry built into the chromosomes long before any of us were born. Scientists estimate that roughly 12% of women on Earth carry this mismatched gene combination. Some studies suggest the number could be higher, but even at the lowest estimate, that's hundreds of millions of women walking the planet right now with the genetic blueprint for an extra dimension of color perception built quietly into their eyes. But here's the catch. Most of them will never know they have it. Having a 4th cone isn't enough. Your brain has to learn to use it. From the moment you're born, you're growing up in a world built for 3 cones. Cameras record in 3 channels, screens project in 3 channels, paint sets, clothing dyes, printer inks, all designed around red, green, and blue. Your visual cortex, the part of your brain that processes sight, wires itself around that system. So even if your retina is sending signals from a 4th cone, the brain, having never learned to distinguish them, treats them as noise and throws them out. Scientists call this silent tetrachromacy. The eyes are fully equipped, the genetics are in place, the 4th receptor is firing, but the extra dimension of color is invisible. Walking around inside someone who doesn't know it's there, out of the tens of millions of women who likely carry the gene, estimates suggest only a fraction of a percent have brains that actually make use of it. The rest see the world the same way you do. Only a handful of humans on the entire planet see the full palette their biology can produce. Proving that someone is a true tetrachromat is brutally difficult. You can't test them on a screen. Screens can only display what a 3-cone eye can see. So asking a 4-cone eye to prove itself on a 3-cone display is mathematically impossible. The screen literally cannot produce the colors a tetrachromat would need to discriminate. Printed color tests don't work either. Ink mixtures fall inside the same 3-cone spectrum, same problem. To identify a genuine tetrachromat, scientists have to build entirely custom experiments. Natural pigments, precise, narrow wavelengths of light projected from filtered lamps, careful statistical analysis across thousands of tiny discrimination tests run over hours, sometimes days. The Cambridge team designed a specific challenge. Subjects had to distinguish between 2 adjacent colored lights that, to a normal trichromat, looked completely identical. Most subjects fail, they guess, they give up. But every once in a while, someone passes, consistently, reliably, across hundreds of trials. Only a handful of women on Earth have ever passed. One of the first confirmed tetrachromats was a doctor from Northern England. In the scientific literature, she's referred to only by a code name, CDA29. When researchers tested her, she scored higher on color discrimination than any human subject they had ever recorded. No one had ever come close. Years later, a second case emerged, a woman named Concetta Antico, an artist living in San Diego. Concetta had spent her entire life painting the world she saw, never quite understanding why other people didn't find her paintings obvious. Her canvases glowed with color variations that to 3-cone eyes looked almost exaggerated. Rocks with streaks of pink and violet, leaves with 12 distinct greens, shadows that weren't gray at all, but blue, amber, and lavender layered on top of each other. But to her, they were documentary, accurate, a simple record of what she'd always seen. Her paintings, in a strange way, became a map of a visual world that exists in plain sight, but had been hiding from the rest of us the whole time. So what does a tetrachromat actually see? Not new primary colors, nothing alien or psychedelic, nothing you'd recognize as different if you compared notes side by side. They see between the colors the rest of us see. A green leaf isn't one shade of green to them, it's a dozen, each one distinct, each telling its own subtle story about light and water and pigment. A sunset isn't a smooth gradient, it's a stack of separate ribbons, layered and visible all at once. The bark on a tree has color. A piece of fabric in low light reveals warmth or coolness most of us would simply call gray. And the strange part is, they often can't describe it to us. The language of color we share evolved for 3 cones. Their experience extends beyond the words we have for it. Imagine if overnight you could suddenly hear a new octave of music. Not louder, not stranger, just more. The notes were always there, you just never had the ears for them.
Tetrachromacy is just one story in a much larger map, the quiet ways your DNA shapes what you perceive, remember, and feel. On Human Decoded, we're mapping all of it. If you want to know what else your biology is doing behind your eyes, subscribe.